Hello, and welcome to our next lecture in the Microbial Genetics series of lectures. And the overarching theme of these next few lectures and videos are the functions of genetic material. Okay, and we talked about this a few times when I mentioned the central dogma, right? And we talked about it when we learned about retroviruses, how the retroviruses go against the central dogma, and that's because they start as RNA and retroactively go to DNA, then RNA to protein. Okay. So even though they break the cycle, they still end up using the DNA, RNA, protein motif known as our central dogma. Okay? And I say know this because this is really important and the real crux of everything in viruses and in microbial genetics. Okay, and what do we mean? It means DNA is transcribed into RNA. RNA is translated into protein. Protein does the work. Okay, DNA, RNA, protein. And remember these terms, transcription and translation. Get into those. But transcription is essentially, like its name implies, transcribing something, writing something across, right? So we're just passing off the information. We're going to get out of the nucleus if we're a eukaryote. We're going to just get it out. Okay, so we need a messenger for that. Even, there's always a messenger, and DNA uses RNA as its messenger. In fact, we even call it the mRNA. In the last video, we mentioned messenger RNA. That messenger RNA is translated completely different now. We're not even in nucleic acid, so that's why we're translating it into protein. Okay? And that's going to be going from nucleic acid that we learned in the biochemistry chapter into protein that we also learned about. Okay, so what's the whole idea behind this, right? How we enact this. The idea is that we have a genotype. What is a genotype? A genotype is the actual sequence of your DNA, right? And that sequence will tell your whole body what to do. Okay, but how does the same DNA become an eyeball cell and another cell become a liver cell, right, or a brain cell? Okay, and so that's kind of the study of it all. See, we have all the information in each and every cell, but some of that information might not be useful in each and every cell. Okay, and so the genotype actually can only be seen as a phenotype. Okay, and so what we visualize, right, what we see, what we can measure, is called the phenotype. Okay. So getting a brain cell here and getting rods and cones here are based on certain environmental conditions when you're in the womb. Right? So you will start to divide in the womb, and these cells will begin to differentiate with the phenotype. Okay, and rods and cones also replicate to some degree. So the environmental conditions play an important role in phenotype. All right. Then we've talked a lot about DNA replication, but we didn't actually go into how DNA replicates. We got it into it when we talked about bacterial genomes needing to replicate before they can undergo binary fission. We talked about DNA replication when we briefly talked about mitosis. Okay, so we have to grow and divide. In order to divide, we have to have genetic material in each cell. Why? Because otherwise you're not a cell and you're not alive. Not a trick question. But as we proceed, we need to understand how DNA is replicated. Okay, so it's easy to take for granted all the stores of information and Google, ChatGPT, and even this lecture and this nice picture. But these scientists, Mendelssohn and Stahl, had to develop a experiment to actually prove that this is what happens. Okay, so when we say how is DNA replicated, you might see that question on my exam. You might see that question in the future. But more importantly, it's just wanting to share with you how your DNA replicates, right? This is us also, not just microbes. Okay, so we don't have the conservative model of passing on our genes, right? Okay. So we don't take the copy of our DNA, copy it, 1, 2, and then put those new copies together and then keep the original, okay? Very conservative, keep the sanctity of the original template, okay? That does not happen. That would not be energetically efficient, as you'll see, comparatively to our semi-conservative model, okay? Which explains that the DNA is copied strand for strand. As I separate, the strand is copied, goes over here. As this separates over here, the strand is copied and goes over here. Now the two copies are on the other sides. There's an original template on each one and a copy on each one. Okay, that is how it works. Okay, and then those copies will then separate, right? So now this separates over to here, and that one separates over here, and those each get copies, right? And what do I mean by pairing up, right? Remember the A's and the T's and the C's and the G's? They are literally pairing up with a copy of that DNA. And then that means that we're not doing the dispersive model of DNA replication, okay? We don't do it randomly and pass it around. This would imply a very coordinated sequence of events that just seem highly improbable. Okay? So, semi-conservative model, DNA replication. Okay? We alluded to it in the former section. Right? We need the energy, we need the sugar phosphate backbone. In order to do that, in order to add more to a growing strand in DNA replication, right? In order to get that sugar phosphate backbone with nitrogenous base, we need to have 3 phosphate groups. Okay? The 1 phosphate group will remain behind and make up our backbone, but we need the energy of 2 phosphate groups, phosphate leaving groups, in order to catalyze this reaction and get it going, or energetically power it rather. Okay, and this is how it will transfer the energy into sugar phosphate backbone and making that bond. Okay, so that next picture is pretty crazy. I'm going to stop it here and we'll get into the actual page of DNA replication. Just know that we have a copy, we have the master template, okay, and we want to replicate our DNA. We need triphosphates. When we talk about PCR, you'll see we need triphosphates. Okay. PCRs countless times, and you would just get a box of ATPs, you'd get a box of GCPs, GTPs, CTPs, and TTPs. You make them all up, put them in a bottle, and you get them all. So that's how you make DNA. Literally assemble DNA using these triphosphates. All right. Next video, we'll explain DNA replication and it gets a little content heavy, so keep your wits about you.